Hi everyone, and welcome to our first video for our chapter 9. So before we start, just linking everything that we've been doing so far. So we've learned about a lot of things. We've learned about protein synthesis, we've learned about different processes like cellular respiration and photosynthesis, we've learned about the immune system, and these are all really complex and incredible systems, which I'm sure you have figured out. But have we had them forever? How did they develop and why did they develop? So this outcome is really going to bring it all together, and it's going to allow us to learn about what has made us or human beings, and what makes us different to other species, and how we've developed these really complex processes over time, and what has made our species so successful. So this last area of study is going to be on evolution. So the dot point that we're going to be covering in this video is looking at how allele frequencies can change. And this is mostly due to mutations, and we're going to be going through the different kinds of mutations that can cause allele frequencies to change in a population. So what do I mean by an allele frequency? So hopefully you remember from year 11 what an allele is. So an allele is basically just an alternative form of a gene. So for instance, in the example that we're looking at here, you can see that we have black sheep and we have white sheep. So the allele that is for hair color has two different variations. It has a black allele and it has a white allele. And you can see if you look at this sheep, for example, that the black allele is dominant and the white allele is recessive. So that's what an allele is. So what a gene pool is the complete set of an alleles that is in a population. And we can represent how many alleles are in a population by calculating something called an allelic frequency, which is basically just looking at the percentage of each allele that's present in a population and then looking at how many individuals are also in that population. So you can see here that we have got three different scenarios for this sheep population. We've got black sheep that are homozygous dominant, so they've got two black alleles. We've got black sheep that are heterozygous, where they have one black allele and one white allele. And then we also have sheep that are homozygous recessive, and they are our white sheep. So basically all you need to do to calculate allele frequencies is you just count the number of alleles, you divide it by the total population. So for example, if I wanted to figure out what the allelic frequency was for our capital B, for our black fur, I would count all the capital Bs in this population. So if you go through and count all of them, you will find that there are 12 black alleles in this population. And then do the exact same thing for the white alleles, and you'll find that there are 20 white alleles in the population. We also have 32 sheep in total, so you can see down here the calculation that we would use to figure out the allelic frequencies, and they're the exact same as figuring out population percentages in populations. So for instance, the frequency of the black allele, I'll do 12 divided by 32, which is 37.5%, and then the frequency of the white allele would be 20 divided by 32, which gives us 62.5%. So here you can see that the white allele is actually more prevalent in this population, but because it's recessive, it's often masked by the black allele, but it is more present in this population. So it's really important to be able to understand how to calculate these allelic frequencies, because it does come up a lot, especially when we're looking at genetic diversity of populations, which we will be doing. So the whole reason that we're going through that is because diversity is so important for evolution. So a large and diverse gene pool will contain greater variety of genes and alleles. So for instance, in that last example with the sheep, there was only 2 alleles present, but in more diverse gene pools, we're going to have way more alleles present. So we might have white sheep, black sheep, brown sheep, spotty sheep, all different kinds of sheep, which is a diverse gene pool. We really want diversity in our gene pools, because diversity leads to a greater number of genotypes and therefore phenotypes, which are physical characteristics of an organism. And genetic diversity is really important for survival. So why would we want all this diversity? Well, let's say that a disease wipes through a particular bird population, for instance, and some birds have an allele that protects them from that disease. Okay, so the more diversity that we have in our populations, the more likely we are to have characteristics that are going to be protective and are going to allow the population to survive despite adversity around them. So having diversity in the gene pool doesn't just happen. There's something that needs to happen first for diversity to occur. So the only way that alleles can change and new characteristics can develop is through mutations. So a mutation is a permanent change to a DNA sequence. Now we have already, way back in Area Study 1, we learnt about the genetic code and DNA sequences and how to read them. But just for a quick scratch up, so these are our nitrogenous bases that we would have learnt about already. So we have adenine and thymine, which pair up together, and we have cytosine and guanine, which pair up together. So we looked a lot at these DNA sequences. We looked at how to transcribe them and translate them into proteins. And hopefully you notice that the order of these of this DNA code is really, really important. We know this is important because this genetic code is making proteins. So if there is a change of order or a change of nucleotide or anything in this DNA sequence, that means there's going to be a change in the mRNA produced, and therefore there's going to be a change in the amino acid sequence that's produced. And if there's even one change in that amino acid sequence, the protein shape can be heavily affected. And we know that protein shape, especially in enzymes, is really, really important for its function. So what mutations do is they will change the genetic sequence in some way, which is therefore going to change the protein that is made at the end. So obviously this can be good or this can be bad. So there might be a change in the protein structure that is advantageous. So this might lead to the production of a protein that actually enhances survival. So it might enhance, the action of a certain enzyme or something like that. We also have neutral ones, so this has no effect on the protein or the individual. Or we can have deleterious ones as well. So mutations aren't always a good thing for diversity. Sometimes mutations can lead to an abnormal function of a protein, which might really impact the survival of a group or an individual or a population. So mutations can actually also hinder the survival depending on how they change the protein. But what we're looking for evolution and the survival of species is we're looking for these advantageous mutations. So it's really important to note that mutations are the only way that new alleles can be introduced into a population, and therefore they're the only way that genetic diversity can be either increased or decreased. So it's also important to note that these mutations, mutations happen all the time, but it's mutations in germline cells that are the important ones for evolution. So if we have a mutation in a somatic cell, which is just a normal body cell, that can't be inherited in the next generation, and therefore that mutation doesn't affect the offspring. Whereas if there's a mutation in a germline cell, which are the cells like sperm and egg that produce offspring, then that mutation is going to be inherited, and that's how we start to see changes in allele frequencies and in characteristics as well. So mutation is the main driver of this genetic diversity and natural selection. So there's a variety of different mutations that we're going to be looking at, but the main one we're going to look at are things called point mutations. So point mutations just mean that there is a change in one single point in the DNA sequence. So there's a change to a single nucleotide in a gene. So we're going to further split these down into three different or four different types as well. So we're going to split these down into silent, missense, nonsense, and frame shift mutations as well. But basically point mutations are mostly substitution mutations. So this means that we might have a DNA sequence that looks like this. And a substitution mutation is going to mean that a nucleotide pair is going to be substituted for something different. So let's say that this A accidentally gets, turned into a G. So that is completely changed this piece of DNA. So that's what a substitution mutation is. So it's substituting one nucleotide pair for another nucleotide pair. We can also have additions or deletions. So this is when, for instance, we've got this same DNA structure here, but instead of replacing this with something else, it's completely deleted or removed. So you can see that this would have a massive impact because all of the bases that are upstream or downstream of that are going to be shifted, and that's what we call a frame shift mutation. Additionally, we can also have the addition of a nucleotide pair, which again can cause that frame shift mutation. So here is a look at all of those with pictures, which will be a lot more helpful to explain this concept. So at the start here, we've got our normal gene. So this is a normal gene, nothing's happened to it. You can see the sequence here, AAA, ATG, CTT, CTC. And then you can also see the mRNA that's produced from that template strand, and then the corresponding amino acid sequence that those codons code for. So that's what the normal genes should produce. When we have point mutations, we can split them up into a lot of different categories. So you can see here in this one, we've had a substitution. So this base pair here of T and A has been substituted instead for a C. So our T has been swapped out for a C. So what this has done is it has changed this codon here. So the genetic sequence is different, but as you can see down here, the amino acid is the same. So we call this a silent mutation. So this is when there is a change to the DNA sequence, but that change in DNA sequence has not actually changed the amino acid that it's coded for. So this can be possible because the genetic code is degenerate. So what that means is that one amino acid can be coded for by many different DNA sequences. So if you remember the codon table that we used, one amino acid can be coded for by a range of different codons. So the degeneracy of the genetic code allows for silent mutations to occur, and in this case the amino acid hasn't changed, and therefore the gene hasn't changed, so this is a silent mutation. We can also have a missense mutation. So again, you can see in this one, we have had a substitution from our C cytosine base here to a thymine base over here. So that has changed the codon from CTT to TTT. You can see in this one that this change in this codon has actually resulted in a change in amino acid. So this is what we call a missense mutation. So this is when the codon, the change in the DNA sequence has resulted in a change of one amino acid in the sequence. And depending on what amino acid this was, or what role this had in the structure of the protein, this could either be advantageous, it could be deleterious, it could be a really bad, it could be part of the active site, or it could be neutral. It's hard to say until you've actually seen the protein. And then lastly, we have a nonsense mutation. So nonsense mutation is where, again, we're substituting a base. So in this scenario, we're changing our guanine to a cytosine. So the codon is changing from ATG to ATC. And you can see here that particular codon that is then transcribed in the mRNA sequence now codes for a stop codon. So that is what a nonsense mutation is. It's when the change in the DNA has resulted in a stop codon. And you can, this is probably the most impactful mutation that can happen, because as you can see, if there is a premature stop codon, that means that the protein is prematurely stopped, so it doesn't continue on making. So that's going to cut the protein or the amino acid sequence really short. So a stop codon is normally going to significantly impact the structure of the protein. We then come over here and we have our frame shift mutations. So this is when we can either add or delete a whole base from the genetic sequence. So as you can see over here, we've got our, we're still working with this normal gene up here, but we have added this entire base pair sequence. So we've added a cytosine guanine pairing, And you can see that this has actually shifted the whole reading frame back, because we know that our codons are read in threes. So if we insert a base here, that has changed the triplets that are being read, and therefore it changes every single codon that goes after this one. So you can see here that every single codon and therefore every single amino acid that's being coded for is changed from this point. So a frameshift mutation can have a severe impact on the protein structure. A deletion is very similar. So in this case, we have deleted a base. So in this one, we have deleted our adenine uracil pairing. And you can see that that has also shifted the frame. So again, we read in threes. So this has changed the codons that are being read, and therefore has changed the amino acids that are being produced from that point. So frameshift mutations have the most impact on a protein structure and are the most likely to change an organism's phenotype as well. So these are all the different kinds of point mutations that can occur from substitutions or insertions and deletions. We can also have block mutations. So these are much bigger. So these involve chopping and changing much larger sections of DNA. So this is about the chromosome level. So you can see here that at the chromosome level, we can have whole sections of DNA be deleted. So this whole section of DNA in the chromosome has been deleted. We can have duplications. So, when the chromosomes are, replicating and things like that, we can have a duplication occur. So here you can see that this gene has been duplicated twice over, which has made 2 copies of that genetic information. We can have an inversion. So an inversion, it just means basically flipping the gene, flipping it back to front so it's sitting backwards, essentially. And then we can have a translocation. So a translocation is when we are taking a section of DNA from one chromosome and it's actually being added to a different chromosome. So all of these mutations usually occur in meiosis, because that's when we are, splitting our chromosomes, and this is where most of our chromosome, replication happens. So this normally happens then. And you can imagine that this has severe consequences on the DNA sequence of an organism, the proteins that it produces. This would have a massive impact. So these sorts of things can also lead to diversity and changes in organisms. The last one we're going to talk about here is aneuploids and polyploids. So these ones are probably the not as likely to be seen in humans, but they still can happen. So these are mutations that affect entire genomes. So we've gone from the single, DNA nucleotide level to the chromosome level, and now we're on the entire genome level. So when we have a loss or a gain of a single chromosome, we call this aneuploidy. So this is a chromosomal abnormality in which an organism has the wrong number of chromosomes due to an addition or a loss. So an example of this that you're probably really familiar with is Down syndrome. So people with Down syndrome, if we look at this middle one here, so this one here is a typical person's chromosomes. This middle one here is someone with Down syndrome. So you can see that they have an addition of the chromosome 21. So they now have 3 copies of chromosome 21. So that's what aneuploidy is. It's the additional deletion of an individual chromosome. We then have polyploidy. So polyploidy you don't really see in humans. Unfortunately, humans with this sort of thing typically don't survive very long, but you do see a lot of polyploidy in plants. So polyploidy refers to a chromosomal abnormality in which an organism has an incorrect number of a whole set of chromosomes. So for instance, this one on the end is the chromosomes of a watermelon. So watermelon actually have 3 sets of every single chromosome instead of the usual 2. So polyploidy is working with entire sets of chromosomes. And again, like I said, not common in humans, but can be more common in plants. So thank you for listening. If you have any questions, bring them to class, and I will see you there.